So, good evening, dear students.
Good evening, sir.
Just wait.
Good evening, sir.
So, today we will discuss. Already yesterday also we discussed some few kinds of Cercospora. So, classification of Cercospora. So, the division is Deuteromycotina. Class is Hymonocetes. Family is Pratulaceae. Genus is Cercospora. So here, just wait. So there are about 700 species. The genus is Cercospora, which causes leaf spot diseases. What is this? Leaf spot diseases. In this, it is the disease or leaf spot disease only the various host belonging to the cells, pulses, oil seeds, fibers, crops, fiber crops, and vegetables also. In most of the Cercospora, the species, the Cercospora personata, Cercospora arachidilocola, which is the generally this disease called as Ticca disease of groundnut. What is the scientific name of groundnut?
Arachis hypogaea.
Arachis hypogaea. This mostly it is seen in the groundnut. It is Ticca disease. The mycelium consists of a multicellular, separate, and branched hyphae. So inside of the host forms it is lobed hastoria, which penetrate the cells. So what are the symptoms? The symptoms of this Cercospora disease in the groundnut. The Cercospora personata and Cercospora Archidicola cause leaf spot disease in the groundnut, which is popularly known as Ticca disease. Popularly known as Ticca disease, right? So it serves cases stems may also be affected. So in the initial stage, it is difficult to distinguish the spores produced by the two species. The two species is Cercospora personata. It produces spores which are usually circular in the shape, dark brown in color. Just look at slide. My slide is visible.
Yes, sir.
So what is the leaf? This one. Yes.
Groundnut, sir.
Yes. This is the groundnut. How it is look like? The spots is how it is look.
Brownish and yellowish, sir.
Brownish and
Surroundings are yellowish, sir.
yes. So the male spots is it is circular in shape, dark brown in color. So the yellow is the not spots. When the dark spots is started on the surface of the leaf, automatically the loss of chlorophyll. The loss of chlorophyll automatically it turn to the yellow color. So the spot is in the leguminaceae, these plant Arachis hypogaea groundnut plant. It is the dark brown color. So this method it is up to 8 millimeters. 8 millimeters in diameters. So the spots produced by the Cercospora personata and Cercospora aracheticola are irregular to the circular. Irregular to the circular. These are much bigger in the dimension. How it is look like? It is very big size. So it is day by day it is increase. Okay. So have a tendency to call the spots are surrounded by the yellow halo. It is day by day the dark brown spot is bigger. Then it is the spots are surrounded by the yellow color, yellow halo, and later turn into the reddish brown. It is reddish brown to black in color. So how many colors here? The formation of the first of all, when the Cercospora disease attacked this plant, the first appear spot is yes. Tell me. Yes, what happened? Tell me. The first is the dark brown color, and it is yellow color. Later, the yellow color it turn into the reddish brown. Then this reddish brown turn to the finally black color. Yes, my voice is audible. Do you understand?
Yes, sir. We have understood.
The first, what are the first color?
Brown. So it's a brown, surrounds with a yellow color. Then it
Yes.
leads to the reddish brown, and finally it turns into black color, sir.
Black color. This is the way of the when the Cercospora or Ticca disease attacked to the this Arachis hypogaea like groundnut plant. This so the spots appear these first of all brown color, then it is yellow color, yellow to reddish brown to black color. Oh, this is to confirmed with these symptoms. This is Cercospora or Ticca disease. So then we will go for the how to control this fungi. Then we will go for the pesticide shops, we will use the pest management. So that is different story. So these are the symptoms. So in your practical, you take this type of diseased leaf, you cut it with the along with the pith means two potato pieces. The middle of the two potatoes piece, this leaf also inserted. Then we'll cut the thin sections. After you get the thin sections, then you observe under the microscope. So here, what are the symptoms? These also some spores, conidias or conidiophores. So you find out the conidiophores. You will show the examination in front of the examiner, the practical examiner. You will show. They will give the marks. Okay. Compulsory. Everyone must and should show the disease. After disease symptom, after getting the sections, you observe some conidiophores. What are the diseases? The cercospora disease or ticca disease. It is entered to this leaf. Their life cycle completed in this leaf. What are the sexual reproduction? What are the asexual reproduction? Suppose the sexual reproduction, it produce the conidias and conidiophores. Just these conidiophores and conidias will show the examiner. Okay. So that's why what we called in this leaf spot of groundnut or ticca disease of groundnut or cercospora disease of groundnut. How many names? Leaf spot. Yes, leaf spot disease.
Cercospora disease and also ticca disease.
Cercospora disease and ticca disease.
And very, very popular, the name is ticca disease in groundnut, right?
Yes.
So here, we will observe the cross section. So I will show the diagram. Just wait. So your right side, this slide is visible.
Yes, sir.
So right side. So here, when cross section of this diseased groundnut leaf, the first one is the infected leaf. You observe the diagram. A, it is the leaf spot disease of the groundnut, and then the asexual fruiting body of cercospora is called acervulus. Asexual fruiting body of the cercospora is called acervulus. Acervulus. The mature hyphae, which are brown in color, are aggregate to form a stroma near the stomatal cavity of the leaf. So the conidiophores arise in groups from the stroma. The conidiophores are in dark in color, small, unbranched, and generally aseptate. They are thicker than the rest of the hyphae. So the conidia are produced acrogenously. Each conidium is long, cylindrical to obclavate. Obclavate means rounded at the base and tapering towards the end. And the color ranges from the highline to ashy gray to in the light brown color. When after the taking of the section, cross section. So here, these are the symptoms. Just you observe. So here the B, these are the conidiophores. What are the conidiophores? Already we discussed in the conidiophores. it is a body-like structures. These are the conidiophores. In the upper side, it is conidio. You know difference between these two? These are the host tissue, stroma and substomatal chamber. The C is the acerulus. The acerulus, what we call acerulus, it is the asexual fruiting body. So this is the below part is the stroma. It is the conidiophores. It is the conidium or conidia. The conidia are unbranched. Okay. So these are the entire conidial structure. It is the conidia. Do you understand? Yes?
Yes, sir.
So these simple, only in this Cercospora, you write your symptoms, then the cross section of acerulus. So the cross section of acerulus, you observe in the stroma in the substomatal chamber, it is conidia and conidiophores. And after you take in the cross section, just you observe these conidia and conidiophores. Yes, conidia and conidiophores is unbranched. This will you show the examiner at the time of your practical examination. Okay, and already we discussed in the symptoms. So this is the regarding about the Cercospora. Disease in the groundnut, Ticca disease in the groundnut, leaf spot disease in the groundnut. Right? Clear?
Sir, if conidia undergoes germination, what will it produce, sir?
Yes. Once again.
If this conidia
yes.
will germinate, what it will produce, sir? Like another.
Yes.
I'm having a doubt, sir. Like what conidia after germination will produce?
Automatically, new mycelium, na?
Okay,
same,
sir.
New mycelium. In every, this is the last stage. Then the conidia is some breaking of the suppose cell wall or anything. They are different, like wind or water, whatever may be. Through the wind, the breakage of the cell wall of this one, they are in suitable and favorable condition. Some of the fungi, favorable condition, they germinate the newly Cercospora. Cercospora is the one type of fungus.
Okay, sir.
These conidia detached from the conidiophore, it is released by any type
like
of
air, water, anything else.
Whatever may be. Its suitable conditions are favorable conditions are there. It is immediately develop a Cercospora fungi. Again, he entered to the mostly in the cereals. Yes, it is mostly in the cereals and some of the pulses, oil seed. When after detached from the conidiophore, these conidia, for example, this will go for the suppose in the groundnut seed. It is dormant, dormant period. When suppose the farmers cultivate these seeds of the, then they are providing some water, some of the nutrients from the soil. Then it is immediately germinate. It is germinate starts when the favorable conditions. It automatically germinate the new Cercospora. Do you understand? Any fungus,
yes,
this one. Some of the fungus, unfavorable conditions, the different ways. Some of the, already we discussed in the previous class, some of the, some conidian, the end, like what we called in the end stage. The end stage, like conidia, whatever, juice spores or any spores, sometimes they are in the dormant period, like few hours, few days, few years, whatever maybe. That depends upon the fungal
condition.
Depends on the fungal condition.
Yes, sir.
So it is immediately favorable condition. It is immediately germinate. There is no germination condition. It takes the long time in the dormant period, right?
Yes, sir.
So that is the germination process. So for example, vegetative reproduction. What happening? All the fungus mostly it is the fragmentation. Some of the yesterday we yeast budding. Detach the mother cell, it is immediately grow the new
as a new individual, sir.
New individual. Same like this, but it is the conidia needs, the fungus needs some favorable condition.
Okay,
then
sir.
Immediately it grows. Depends upon the fungus. Some of the fungus, the dormant period is very less. Some of the fungus dormant periods is very low. So on that's why some seeds also, generally not only fungus, some seeds also. Suppose sowing today, for example, some seeds sowing, it is next day it is immediately germinated. Some seeds 5 days, some seeds 1 week. So for example, is turmeric is there, that is rhizome, not seed. So it takes 1 month of time. Suppose cotton seed, it is like 1 week of time. Suppose rice, paddy, it is 3-4 days of time. So depends upon the, these are the, so that's why the farmers, they are using some artificially pesticides. They immediately, suppose some of the seeds, it is very long period of the dormant. So the breaking the dormancy, some scarification, some chemical treatment, some gibberellic acid treatment,
Yes,
some
sir.
hormone treatments are there. So some treatments are there, it is immediately grow. So that is the, so these are the very important in this the definite questions in the, your practical 100% is question. In practical, it as a major experiment. You take the disease leaf of the groundnut, then you do the cross section. You observe the under microscope. These you identify the these conidia force. Just you will show the examiner. So first of all, you write in the practical exam classification, some introduction of 2-3 species of these regarding this one. So some, what are the symptoms in the groundnut? Then what you observe in the under the microscope, and diseased infected leaves of groundnut. Oh, this is the ticka disease symptoms. It is
Okay, sir.
first of all dark brown color, then it is yellow color, then it is reddish brown, it is to the finally black in color. These are the process. Okay. So what happening in the life cycle? How it is completed life cycle in the groundnut, right? So here another one is these are very important. So generally, according to our syllabus, the groundnut is a very popular. So Cercospora leaf spot in the sugar beet also is there. Same like just you observe the spots. So this is also some Cercospora leaf blight of soybean also. So just observe, these are the conidias, conidia of Cercospora. Just so how to control this Cercospora disease? Some crop rotation, early planting, choose later the maturing cultivars, chemical control. The chemical control, a foliar fungicide application may be applied to the early pod development. So any stages of any plant like cereal plants, pulses in the oil seeds plant caused by these diseases. So use the fungicides. The fungicides is available in the market. Is very different types of fungicides are there. Any fungicide you use this one. So next, so this is the groundnut leaf spot, and it is also called the early leaf spot of the. It is the abaxial surface, the groundnut leaflets. So how it is going on? Just these are the structure of conidia, morphology of conidiophores and conidia. This below part is the called is the for example A Cercospora conidiophores. B is the person tonight. These are the B. This one is the unbranched. This is the conidia. So how it is just you observe this one. This is the leaf infection of the host tissue. These are the infected leaf. This is the mycelium without Hastoria. The early leaf spots of symptoms, upper leaf surface. You know upper leaf and lower leaf. So here, these are the conidiophores on the leaf surface. So the lower leaf surface. So here routine practice, some fungicides are Bordax mixture, dithen, Fical, Bristol, copper sulfate mixture. These are the many types of disease cells. So coming to another one is so same like this Colitotrichum. Just wait, I will share the Colitotrichum nodes.
రెండు రాసావా పశ్చిమ ಪಾಕಿಸ್ತಾನದ ಕಡೆಗೆ ನುಗ್ಗಿದರು ಇದೇಂటి ఇది తప్పు ఉంది కదా సేనా కుడి తుకడి
Mute yourself.
Yes.
Okay.
ರಾಜಸ್ಥಾನ ಮತ್ತು ರಾಜಸ್ಥಾನ ಮತ್ತು ಕಚ್ಚದ ದುರ್ಗಮ ಪ್ರದೇಶಗಳಲ್ಲಿ
ఇది 10 రూపీస్ అవసరమా దానికి పృథ్వీ క్లాస్
Hello, sir.
Hello. Yes.
Yes, sir.
Actually, suddenly
Internet connection problem, so it might be.
So the classification of Colitotrichum, the division is Deuteromycotina, Coleomycetes, and family Melanoconials, Colitotrichum. So all the species of Colitotrichum are parasitic and higher plants such as sugarcane, cotton, beans, onions also. So red rot of sugarcane. So red rot disease in the especially we seen in the sugarcane. Red rot of sugarcane. So when suppose in the sugarcane stick is sugarcane stick is there, just it is open the sugarcane, the two parts. Sometimes we observe in the red color, it is appeared look like the red color, that is disease, that is red rot of sugarcane. What is this? Red rot of
Sugarcane.
So it is an important disease and is caused by the Colitotrichum falcatum. Colitotrichum falcatum. Colitotrichum. What is the disease name is? The host. Colitotrichum falcatum.
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 Falcatum. Yes.
This disease is quite especially in eastern Uttar Pradesh and Bihar. Very dangerous in this. The most of the charaku cultivated in the Uttar Pradesh and Bihar also. So it is once attack the disease, it is growing the entire within the short time. This disease spreading in the rapidly. So then what happening? There is no production of sugar. There is no production of sugarcane byproducts like jaggery and something. So the mycelium is either intracellular. The hyphae are freely branched, separate, colorless, and contain characteristics of oil droplets. Oil droplets. So here the Colitotrichum is a parasitic in higher plants, in especially sugarcane, cotton,
beans, and onions.
So mostly it is dominantly we observed in the sugarcane. How it is appear? It is the red rot. It is look like red rods. So it is called as the red rot of sugarcane. So it is caused by this disease, caused by the, what is the fungus name is? Colitotrichum falcatum. Falcatum. So here, especially in the eastern Uttar Pradesh and Bihar, the mycelium either inter or intracellular, the hyphae are freely branched, separate, colorless, and contain characteristics of oil droplets. So we'll go for the symptoms. What are the symptoms? So the Colitotrichum falcatum causes red rot of sugarcane. The red rot of the fungus generally attacks in the stems and midribs of the leaves. Midribs of the leaves. So here, look here, this slide. And the plant growth is adversely affected when the Colitotrichum falcatum attack to the sugarcane. The plant growth is affected. There is no plant growth. So in this, it is longitudinally wrinkles are formed. The affected sugar canes, show a considerable loss in the weight and it is become lighter. Generally, when cut the sugar cane after maturity of sugar cane, when cut, it is like how it is it is healthy sugarcane. It like some 250 grams or 300 grams. When this disease attacked to the sugarcane, when cutted the sugarcane, it is look like, suppose it is very long of sugarcane, the weight is very less, like 100 grams, because the fungus absorb all the nutrients from the sugarcane. So that's the reason the cane is become loss of the weight, right? So the affected stems, when split longitudinally, show the red streaks. So just you see these diagram. Yes, you observe.
Yes,
Yes,
sir.
sir.
So these are the red streaks. How it is affected in the Polytotrichum falcatum. These are the red streaks in the internally and externally. How it is observed. Internal discoloration with the prominent pits, varying interstates of the rind discoloration. Though it is the external part, this is the internal part. These are the spots, serial spots of the red spots. So the lower leaves, red longitudinal, it is also affected leaves also. So red longitudinal bands are seen and along with the midrib of the leaf. You know midrib, the center part. How it is
Yes,
the
sir.
leaf,
Yes.
like maize, you know, maize leaf, same like that. So in the middle part, middle vein, that is the midrib. So then sugarcane leaf, it is midrib also affected due to this diseases. So the upper leaves turn pale, drop down slightly and whitener. So the midrib part is the some bonds, longitudinal bonds in the upper leaves turn in the pale color. It is drop down slightly and it is whiter, right? So here, so it is a serious and destructive diseases. The first reported in 1893 from the Indonesia, and in India first reported in the 1901 in the Andhra Pradesh. Then the disease epidemic occurred in the 1939 to 1940 UP and Bihar. So disease is soil and seed-borne diseases. The fungus is the how it is entered into the plant through the soil and seed-borne. So distribution in the tropical and subtropical region of the world. In India, it occurs most the sugarcane growing states, particularly Bihar, Uttar Pradesh, Madhya Pradesh, Haryana, and Punjab. So in Telangana, you know Telangana, what are the which are the district cultivated this sugarcane? Anyone? The sugar factory, where it is located in the sugar factory, Nizam Sugar Factory.
Nizamabad.
Yes, anyone?
Nizamabad.
Adilabad.
Hmm?
Bodhan, sir.
Yes.
Bodhan. Bodhan and Medak.
That maybe.
So, which district highest cultivation of the sugarcane in Telangana? Yes.
Yes, sir.
All of you, you are only. Are you belong to the Hyderabad only or any other district?
Other all districts.
Hyderabad.
No. What
No,
about
sir. Kamareddy district.
Hyderabad.
no Hyderabad?
Mahabub Nagar. Islamabad.
Kamareddy district.
Narayanpet.
Kamareddy region, sir.
It is Nizam Sugar Factory. It is located in the Nizamabad district. It is the headquarter. So now it is some differentiated some district now. So it is in the
In
old
Bodhan,
district,
sir.
Nizamabad. So
Nizamabad district, Bodhan, sugar
okay.
factory.
Yes. So most of the farmers cultivated in the Medak as well as the in Nizamabad, farmers also cultivated in this sugar canes. So okay, coming to, because this is the some GK purpose, I ask this question. So next, the main important of the symptoms. How symptoms So after rainy season, the plant growth stop and sugar formation is start. Yes. After plant growth, so some vegetative growth is there now. Every plant in the starting time is the vegetative parts, like the branches, the produce of branches, twigs. That is the vegetative. Once the vegetative growth is stopped, then the sugar formation is start. In this, the symptoms are yellowing and drooping of the upper leaves near of the tip. Later, the infected stems. Then the rind loses the bright color. It become a wrinkled. So cane means sugar cane becomes lightweight and can be easily broken. Already I told you. So generally the healthy sugar cane in long, for example, 10 centimeters or 10 meters or 5 meters, 3 meters, depends upon the variety of sugar canes. When cut it in the healthy sugar cane, it like 250 grams, 300 grams, 400 grams. But when attacked with these diseases, it lightweight and can be easily, it is broken. So like this third one, just you observe the third one, this part. So, and longitudinal ridging of the normally white internal tissue with internal transfer. The white spots are internodes. It is discoloration that may be extended through many joints of the stalk. You know, sugarcane joints, these are the sugarcane joints. Yes, you observe this one. This is the internal part, but this is the external part. This one. So the disease blood red license with dark margins develop in the leaves. That is called as the acervuli. These are the what we call acervuli. The acervuli is the asexual fruit body of the Colletotrichum. In competitive exams, many times asking this question. The asexual fruit body of Colletotrichum. The answer is, what is the answer? Yes?
Acervuli.
Yes, this is the answer is very, very important. Otherwise, the acervuli we are seen in the which stage of Colletotrichum? It is asexual fruit body. We are observed in this in the acervuli. So in the late season, dark dot-like structures, valvity in the texture appear in the shriveled areas near the nodes and the internodes. So desiccates in the small midrib license. These are the midrib. So you observe these parts. It is the midrib. And these continuous lessons on the midrib. Then the starting here, one spot and one spot. Then the entire these sugarcane leaf, it is like this, entirely damaged. So effect of the disease, just you see the finally the disease is very dangerous situation. It is very very dangerous situation. There is no production of sugar. So like this, the entire sugarcane, entire the formation of this one, then broken out. It is very the weight of loss. So these are the this is the leaf spots. The leaves appears this types of part. This is the midrib. So how to start the spots in the sugarcane? It is the cane. It is the sugarcane. This is the sugarcane leaf. That's why what we call it is the red rot of sugarcane. This is the red rot of sugarcane. So effect of disease, the induces conversion of sucrose into glucose and alcohol in a mature cane sugar plant due to the enzymatic action of pathogen. So after enter this the fungus pathogen, it is some activity, enzymatic action. So loss in sucrose content may be as a high as a 33% of the sugar sucrose, right? So juice of bad odor, the juice So the juice, when sugarcane juice, they are the selling person, they are affected or diseased sugarcane. They prepare the juice. The juice is the smell is very bad. It not set well on the boiling due to the conversion of sucrose into glucose. So low quality of the sugar as well as a reduction in the production also. So how to the buyers and sellers, they are in the market. When this, after cultivation of the sugarcane, we will go to the some factories, sugar factories. The sugar who buying these sugar canes, they are estimate the quality of the sugar, production of the sugar depends upon they are only much rate in the healthy sugar canes only. In case they are observe this type of spots and this type of spots on the surface of the sugar cane and surface of the leaves, they are not buying. They are very spend less amount. So the low quality sugar as well the reduction of this disease in it is affected in the production also. So even threatens to disrupt to the entire crop of the field, leading to enormous loss. So the causal organism is Colletotrichum falcatum, Glomella tucumensis. The Colletotrichum falcatum is very, very important, is commonly observed. So these are the systemic position already I discussed. Kingdom Mycota, division U Mycota, subdivision Deuteromycotina, class Coleomycetes, order Melanocoleus, family Melanocaceae, genus Colletotrichum, species the falcatum. The pathogen characters. It is a causal agent. Colletotrichum falcatum is a from class fungi. It is an imperfect fungi. Already I told you what is the imperfect fungi. So the perfect or sexual stage of the fungus has also been described the Golmerella tucumensis. The asexual fruit bodies are called acervuli. The acervuli are black valvety bodies which develop in the clusters. The pathogen survive in the seed cans infected the stubbles, debris, and the soil of the chlamydospores. The chlamydospores are terminal or intercalary. The disease seed and soil birth. The disease is already I told you, it is the seed-borne disease or soil-borne disease. The mycelium of red diseases. The fungus of mycelium is found in the within and intercellular spaces between the pith cell of the host. The hyphae consisting in the mycelium are both inter and intracellular. They are slender, colorless, branched, and septate. The hyphae produce a large number of chlamydospores in the pith parenchyma. So, epidemiology. Mode of spread, how it is spread of the disease. In the rainy season, the disease spread in the very fast and the whole crop dries up. The primary transmission through the soil and diseased sets. The primary inoculums, old fragments of stalk and leaves on which the fungus grow saprophytically. So this is the reason we advise to the farmers. The crop rotation is the best treatment of to control this type of diseases, soil-borne diseases. So generally these fungus, it is the saprophytes. So how it is, the whole fragments of the stalk. Already some farmers after cultivation of the sugarcane will cut the some remaining part. In the base part, it is only remains in the soil. The farmers sometimes burning these type of the fragments of stalks. Sometimes they are there is no crop rotation. Again, they are cultivated in the same. Suppose this year cultivated in the sugarcane, in the next year also same. So already some these fungus debris already in the leaves in the soil are old fragment stalks. When the farmers started in the new cultivation of sugarcane, it is in the rainy season, it is immediately rapidly attacked to the sugarcane. So that is the reason. So that is the reason advised to the farmers, the crop rotation is the best treatment. So secondary in column. Inoculums, the conidia produced in the acervally. What is the acervally? It is the asexual fruiting body of the colitotrichum. So developed along with the midrib of the deceased leaves. So these are disseminated by wind, rain, irrigation water, and also by insects. So in winter air, current helps with the spread of the disease. And compared to the rainy season, in the winter also it is very rapidly spread of this disease. The occurrence of the disease in the upper part of canes indicates aerial mode of the dispersal of the incolumns. So mode of infection. Infection occurs through the nodes. Mainly entry points are the leaf scare. Just you observe this, listen carefully. Mode of infection. Infection occurs through the nodes. The main entry points are the leaf scare, growth rings, root, primordial, and buds. The pathogen can also enter to the stalk through the rootlets, growth cracks, and cut ends of the sets. These are the mode of how to infect the plant, healthy plant, this process. The favorable condition of the disease development. The main temperature range of the 29.4 to 31 degree centigrade for the disease development. The pH is 5 to 6. Drought condition during the initial growth of the favorable. Highly humidity. Waterlogged condition of the soil. Disease cycle. The inoculum lives from one growing season to the next on the debris of the diseased plants. The conidia are short-lived, thus play no role in the penetration of the pathogen. The thick-walled chlamydospores and perthesia are considered as the probable means of the survival. The acervally survives active stage 3 to 4 months in the soil. The survival of the mycelium for this limited period sufficient to provide the easy catching of the succeeding the crop, because sugarcane is partially as no dead season. The disease is born on the seed sets, which serve as a chief means of the survival and spread of the disease. The diseased sets sown in the soil sprout into the infected shoots, which soon produce conidia and acervally. The conidia serve as a secondary means of the infection and spread of the diseases. They get detached to the dispersed through the agency of wind, water, insects. On reaching the surface of the healthy sugarcane plants, they germinate immediately in the moisture retained in the enclosing the sheath. So how it is grow? These are the disease cycle of the red rot of sugarcane. So mycelium grows systematically along with the growing canes. It is host infection. It is symptoms appear. Mycelium, the conidia germinate and germinative. This is the secondary infection. Then it will go for the dormant mycelium, the diseased canes. The chlamydospores in the formation of here chlamydospores. Here to here, the mycelium, it is directly chlamydospores. These chlamydospores in the soil along with the diseased plant debris. So here the mycelium directly, the dormant mycelium in the diseased canes, in the two ways, dormant mycelium in the diseased canes already. So the chlamydospores in the soil along with the diseased plant and debris. Then these, it is the chlamydospores in the soil along with the diseased plant, it is germination. Here diseased sets is advantageously sown. Here this phase is the active phase and the dormant phase. Two phases are this. Active phase means immediately it is growth. The dormant phase is the penetration. So then it is germination through the germinative. The enters the growing canes at the seedling stage. It is the soil borne. Already some old debris consist these Colitotrichum falcatum. And also soil also, it is consist of this Colitotrichum falcatum. It enters in the growing canes at the seedling stage, in the starting stage. Then the mycelium grows systematically along with the growing canes. So this is the dormant mycelium in the canes become active. It is the active phase. This is the disease cycle of the red rot of sugar cane. So here, control. How to control? So the control methods and disease controls. The field sanitation. The field sanitation is an important measures to prevent the wild source of the primary inoculum. It consists of the collection and burning of the sugarcane trash in the field. The plantation is resistant cultivars like CO846. These are some varieties most effective. So the use of sound and healthy seed sets, long rotation of crops minimize the soil-borne infection. Crop rotation in 2 to 3 years. Uh, Set the treatments is the carbondism. It is a fungicide 2.5 grams per liter of water for 30 minutes. The hot water treatment 52 degrees centigrade for 8 hours, 54 degrees centigrade for 2 hours. Hot air treatments, it is the 54 degrees for 6 hours. Removal of infected stool. Plant the crop when the conditions are optimal for the rapid germination and maintain the proper soil moisture. So harvest susceptible cultivars before they have passed the peak of maturity. So the practice of crop rotation with an alternative crop, the end of the planting and rotating the cycle. These are the control measures in the disease control. So next, just wait. So these are the some of the how to control model for the controlling the red drop sugarcane, the application of the Bavestin. These are the some pesticides. Land selection, medium high land, well drained, sunny place, use of the red rot resistance varieties of sugarcanes. These are the variety of sugarcanes. The planting material should be collected from the seed from the nursery. So the treatment with the biofungicides, carbendazim, it is a 0.1 percentage. Then it is follow the long furrow method, trends the method for the planting and irrigation, and balanced fertilization like urea 30 to 35 kgs per hectare, TSP. These are the combination of 40 to 45 kg of hectare. The muresh of potash, it is 25 to 30 kg of hectare. So avoid the rotating. So the practice of the crop rotation is very important. These are the some integrated disease management for the control of red grass of sugarcane. So here also here also we observe the conidiophores arranged in the palisade like manner. The conidia are born on the conidiophores. Each conidium is single celled, falcate and halen. So just I will show the how the conidia structure in the sugarcane. So this is the conidias look like in the yes, you observe the this slide is visible.
Yes, sir.
Yes, sir.
So same like this like conidia, how it is after the section cutting of sugarcane diseased part. We will observe the under microscope. These are the conidias. These are the conidia forms. These are the conidia, right? So this is the stem. These are the acer valley. So this is the license. It is the damaged part. So yes, this one good. So this is the like midrib
of the leaf.
yes, midrib of the leaf. Then when the affected the leaf, these symptoms appear like this one also. So it's very just wait. So just you look here, this one, the leaf is affected leaf is right.
Yes, sir.
So then, so finally, what happening here? Just look here. So there is no formation of sugar. So it is becomes a loss of weight. Like some more images. Finally, the entire plants become this one. Yes, observe it. Okay. So, yes. Okay. Yes, this is the healthy, right? Yes.
Yes, sir.
Yes. There is no red spots here. There is no red spots here. The leaves also there is no spots. This is the healthy sugarcane. So then this type of will get more price in the market. So generally the plantation is here like this. Yes. So this is the today class. So we will wait until 7:30. No one please leave the class.
Okay, sir.
So so how many class actually? 20, 21 is the last, and tomorrow also continuation class is there. Just anyone check? Only one class or two classes are there?
One class, sir.
The date is 21.
On 21st, we are having the last class, sir.
Tomorrow there is no class.
Yes, sir.
Tomorrow no class tomorrow.
No class, sir. Only 19, 21.
21.
Okay, fine. So we will cover only in the mushroom cultivation, and regarding mushroom, we will cover the last 21st mushroom cultivation. This is the very
Okay,
little
sir.
bit from how to cultivate the mushroom. So the important mushroom cultivation. So how to the edible mushrooms. So then all the type studies are completed. So I think one more is class is there in the first uh, Candida, Albugo Candida. Just it is very simple. I will finish within the 5 minutes. So I will just wait. It is very simple. Albugo Candida also is very, very important.
So after these online classes, all the we have to come to the college, sir, or it will complete?
No, it will complete. The syllabus is completed. The university, I think within shortly after in the second year classes, I think 27th is the last class, the second years. So they prepare the practical timetable in each paper 4 days. How many papers? 4 papers. Total, how many papers?
4, sir.
4.
Total
Four
4,
papers.
sir.
In each paper, 4 days practical. I think in the last year, before last year, same manner. I don't know this year any changes. I don't know. But last year, before last year, in each paper, entire entire session in the morning, evening, the practical classes must and should you attend. No skip it. There is no option. The dean madam
Okay,
is
sir.
very, very strict. They give the some options like one batch is 2 batches options. So one batch starts this like April month. Some of the batch afterwards, the phase like phase 1, phase 2, like this. So those who are interested in the first batch, according to your timetable, you join the first batch. Madam, all the guidelines they provide the university authorities like Pushpa madam or Kumar sir. But in each paper, 4 days, the attendance is must and should. Any paper. 4 paper.
Okay, sir. When will these practical exams are having, sir?
After 16 days, after all the completion of the 4 paper practical, then immediately they give the timetable. The exams also continue the
Okay,
exam.
sir.
I think
Then
exam.
theory exams, sir?
I think after practical. Yes, any doubts regarding this? So this is the just you observe the this sorry. So how it is? This is the disease caused by the Albuo candida. How it is? The total the leaf is white spotches, right? The classification of Albugo candida, division is Eumycotina, subdivision is Mastigomycotina, class is Eumycetes, order is Peronosporales, family is Albiginaceae, genus is Albugo. It is what we called in the Albugo cystopus, right? So just you observe this disease symptom, the white patches, no? Like this, like this, like this. So the genus Albugo is obligate parasite and attacks several flowering plants belonging to the families, mainly Cruciferae, Compositae, Amarantaceae, and Caparidaceae. The Albugo candida, which also called Cystopus candidus, causes an important disease of crucifers known as, the disease name is white rust, like this. Yes, you observe these diagrams. Yes, are you there?
Yes, sir.
Yes, sir.
What we call the disease name is
White.
white rust.
White rust.
It is usually infects Brassica oleracea, Brassica niagara, Brassica napus, Brassica campestris, and Raphanus sativus. What is the Raphanus sativus?
Mullangi.
Yes, Mullangi. So the mycelium is haline, endophytic, branched, accepted, multinucleate. You know multinucleate?
Yes, sir. More than one nuclear.
Yes. It is button-like Astoria. Generally, we observed in the previous classes, the fungus, it is consist of some fungus, some Astoria. So here, this Albugo candida also, Astoria is there. The Astoria, how it is look like, it is a button-like Astoria. So the examiners ask the questions in the Viva. Which type of or what type of Astoria present in the Albugo candida or Cystopus candidus? What is the answer is?
Button like.
Like
Button-like Astoria present in the Albugo candida, right? So the button-like astoria produced by the mycelium penetrate the living cells of the host to observe the food materials. So it is what we call Albugo candida. It is the obligate parasite. So here, when the asexual reproduction, what we observe, Conidiosporangia and sporangiospores. Conidiosporangia and conidiospores. When we the diseased leaf taken a cross section, we will observe the under the microscopes. What will observe? Conidiosporangia and sporangiospores. Yes, you observe this diagram. This is the host tissue, mycelium, and these are the sporangine basigenous chains. It is the like sporangiophores. So this type of structures we will observe in the diseased white rust diseased in the. What are the families affected in this white rust? Cruciferaceae, Compositae, Amarantaceae, and Caparidaceae. So it is usually the plant. It is very popular plants. Brassica Alaracea, Brassica nigra, Brassica napus, Raphanus sativus. These are the plants mostly affected due to these diseases. The disease name is Albugo candida or Cystopus candidus, and it is also called as white rust fungi. So here two reproductions are there: sexual reproduction and asexual reproductions. So the asexual reproduction takes place by the production of the zoospores inside the sporangia. The epidermis of the hyphae grow luxuriant. It is erect branched and multinucleate sporangiophores. The sporangiophores, it is look like the chain-like structures. Just you observe the diagram, chain-like system. The sporangiospore, their tips are spherical, multinucleate sporangia in a chains. The sporangium is smooth, double-walled, multinucleate, and round in shape. But sometimes it is due to the pressure of during their formation of the results in flattened sides, giving up a polyhedral shape. So generally it is a round shape, but due to the pressure, their formation of results, the flattened sides, so it finally giving up the polyhedral shape. So due to the pressure exerted by the numerous sporangia produced, the epidermis gets ruptured to the sporangia appear in the surface of the leaf as a powdery mass. The white powdery mass, that is the white rust diseases. The sporangia are disseminated by wind or water to other susceptible hosts. In this course, a great percentage of them, the sporangium is directly germinate and produce 4 to 12 zoospores, which are liberated due to the bursting of the cell wall. Each zoospore is kidney-shaped with 2 lateral flagella. In each zoospores, how many flagellas? What are the flagella? 2 flagellas. In the flagella, it consists the lateral. Lateral means sides. The flagella develop in the sides. The zoospores, after swimming for some time, the water present on the surface of the leaf, it germinates by the producing a germ tube which enters to the host through the stomata. Sometimes the sporangium germinates directly without producing the zoospores. This is the regarding about asexual reproduction. We are talking about in the asexual reproduction the formation of zoospores. These are the produced in the sporangiosperms. It is look like the spores are in the chain-like. The chain-like arrangement. It is the round shape. In the sometimes they are pressure. So it is in polyhedral shape also. So here the sporangium directly germinate and produce the 4 to 12 zoospores, right? So it is laterally flagella, and with the help of the flagella, it swims for the water. It is surface of the leaf. This is regarding about asexual reproduction. The sexual reproduction is a oogamous, and sex organs, oogonium and antheridium, are produced in the deeper tissue of petioles and stems. This is sexual reproduction. The sexual reproduction, what are the reproductive parts are reproductive organs or sexual organs in this sexual reproduction of white rust, oogonium and antheridium. Oogonium and it is, listen carefully. Oogonium and antheridium are produced in the deeper tissue of the petioles and stems, where it is located in the petiole and stems. There is no leaf. So the oogonium is the female sex organ. The hypal branch tip swells from the spherical oogonium, which gets cut off from the base of the septum. The oogonium is multinucleate, and the oogonium cytoplasm is differentiated into central ooplasm and peripheral periplasm. The central ooplasm is dense and contains only one egg nucleus. The periplasm lighter and multinucleate. So here the oogonium is the female sex organ. So the oogonium is multinucleate. This cytoplasm is differentiated into the central ooplasm and peripheral periplasm. So here the central ooplasm dense and contains only one egg nucleus. The periplasm is in lighter and multinucleate. These are the regarding about the oogonium as a female sex organ. Next, the male reproductive organ is a multinucleate antheridium. It is formed by the swelling of hypa close to the oogonium into a club-shaped structure. Here in the male reproductive organ is a multinucleadium of antheridium, and it is a club-shaped structure, which is cut off the base by septum. So the antheridium attaches itself the oogonial wall and produces a fertilization tube at the point of the contact. The fertilization tube penetrates through the conidial wall and periplasm and goes deep into the oosphere or egg. One or more mulsi are present in the tube. It is only one of them fuses with the egg nucleus. The zygote or oospores secrete the thick wall around the periplasm. It is takes part in, it is the formation of the sexual reproduction. So finally what happening in the sexual reproduction, the zygote which set free after the decay of the host tissue undergoes a period of rest and produces the numerous zoospores. The zygote bursts and zoospores escapes into the vesicle, which later get dissolved in the zoospores with two lateral flagella are set free. They swim for the some time and infect the subsusceptile host. Here in the sexual reproduction, it is the sexual organs is the oogamous and antheridium. So here the female sex organ is what is the female sex organ? Oogonium. What is the male sex organ is? Antheridium. It is a multinucleate, right? Finally the formation of zygote here, two flagella are there. It is with the help of flagella they swim sometime and infect the susceptible host. Like these already I told you in the beginning, the plants already some of the families affected this white rust, and some of the Brassica species, Brassica oleracea, Brassica napus. Okay, and Raphanus sativum. These plants is susceptible hosts. These are the regarding about Albuga candida diseases. Okay, this is also fungus. What we call it is white rust. So it is also called as Cystopus candida. Right? These are the class. Okay. Next class we will finish our syllabus. The last class, next class we will finish in the mushroom cultivation. The medicinal importance of the mushrooms. We will discuss later. Okay.
Okay,
Any
sir.
doubts? Yes. Any doubts? Thank you.
Thank you, sir.
Okay.
Thank you, sir.
Thank you, sir.
Thank you. Thank you.